All right, everyone. Welcome. To get you ready for your neuroscience exam, we're about to go on a pretty wild ride, a journey that spans 1 billion years. We're going to trace the entire evolution of the nervous system, starting from just a handful of proteins and taking it all the way up to the incredible complexity of the human brain.
Okay, so here's how we're going to tackle this. We'll start at the very beginning with the origin story of the synapse itself. Then we'll see how that led to the first nerve nets, and eventually brains. We'll get into the price of having a big brain, and then ask the big question, does size even matter? Finally, we'll zoom in on what makes our own human cortex so special. It's going to be a great overview.
So, before we dive deep into the nitty-gritty, let's just step back and ask a really basic question. Why? Why do we even have a nervous system in the first place? I mean, what's the point? Really, this is the starting line for our whole evolutionary story.
Okay, let's start our journey at the absolute smallest scale you can imagine. We're talking molecular. We have to go way, way back in time, even before the first neuron ever existed, to find the tiny building blocks that would eventually make everything else possible.
You know, the synapse, this incredibly complex piece of machinery, it didn't just pop into existence, no way. This was a slow burn, an evolutionary project that took almost 1 billion years. It all began with something we call a proto-synapse, which was basically just a molecular toolkit, a box of parts, found in single-celled organisms. From there, we get the aura synapse, the first real prototype, and then, finally, the sophisticated communication hub that we have in our brains today.
And here's where it gets really fascinating. You want to find the ancient history of our own brain? You don't have to look far. Check out baker's yeast. I mean, this simple fungus has about 1/4 of the proteins we find in our own postsynaptic density. Now, obviously yeast doesn't have a brain, but it uses that same molecular toolkit to sense what's going on around it and react. The parts were there from the very beginning.
All right, the next evolutionary upgrade comes from these guys, choanoflagellates. They're still single-celled, but they like to hang out in colonies. And to do that, you need new tools. Suddenly, we see new genes pop up. Genes for things like cadherins and catenins, which are basically molecular glue that helps cells stick together. We also see more advanced signaling proteins. You can see where this is going, right? These are the absolute must-haves if you're ever going to build a multicellular organism with a nervous system.
Okay, now this is a really key point, so pay attention for the exam. Look at sponges. We all know sponges have 0 neurons, right? No nervous system at all. But they already have a ton of the core synaptic parts. They've got orexin, they've got GABA and glutamate receptors, they've got KMQ, all these critical pieces. It's like evolution had all the components laid out on the workbench long before it actually built the machine.
So, the toolkit is ready. The parts are there. Now let's see what happens when evolution finally starts putting them together. We're moving up from the molecular level to see how the very first nervous systems came to be. And they started out as really simple scattered networks.
You can see a really logical progression here, step by step. First up, you get a simple nerve net, like you'd find in a jellyfish. It's just enough to trigger basic muscle contractions, but you can't really steer. Then you get an upgrade, the nerve ring in something like a sea star. This connects the different parts of the net, and suddenly you've got coordinated, directed movement. But the real game changer is when animals develop bilateral symmetry, a distinct front and back. At that point, you absolutely need a central command center. You need a brain to coordinate the left and right sides and move with purpose.
Okay, but here's the catch, and it's a big one. Building a brain, especially a complex one, isn't free, not even close. Think of it as a luxury item. It's one of the most metabolically expensive organs you can possibly have, and that massive energy bill has some really serious consequences for evolution. And this right here shows one of the fundamental splits in the story of brain evolution. On one side, you have warm-blooded animals, endotherms. Their fast metabolism is like having a huge power plant that can fuel a really energy-hungry brain. But on the other side, you have the cold-blooded ectotherms. Their metabolism is much slower, much more efficient, and they simply can't afford the constant energy drain of a big brain. It's a fundamental metabolic budget constraint, and it's why you see this huge difference in brain-to-body size ratios between the two groups.
This brings us to a question that I'm sure you've all thought about. Does brain size matter? I mean, if you can afford the energy for a big brain, does that automatically make you smarter? It seems logical, but let's actually look at the evidence.
It's this idea that bigger is always better, right? We hear it all the time, especially when people talk about intelligence. But is there really any truth to that assumption?
Check out this amazing example from history. You've got two absolute literary giants. Ivan Turgenev, the Russian novelist, his brain was huge, over 2,000 grams, massive. But then you have Anatole France, a Nobel laureate, and his brain was barely half that size, around 1,100 grams. So, right away, this tells you that just weighing the brain isn't going to give you the full picture of someone's genius, not by a long shot.
So, if absolute size is misleading, how can we compare a human brain to, say, an elephant's brain? Well, scientists came up with a clever tool called the encephalization quotient, or EQ. The idea is simple. It's not about how big your brain is in total, but how big it is relative to your body size. It's a way to level the playing field because, of course, a bigger animal needs a bigger brain just to handle all the basic wiring.
Okay, so using EQ does give us a better picture. And no surprise, humans are at the top of the chart. But look at this. The capuchin monkey has a way higher EQ than a chimpanzee. Now, we know chimps are capable of some incredibly complex behaviors, so what gives? This is the proof right here that even a smart measure like EQ doesn't tell the whole story. Relative size is part of the puzzle for sure, but it's not the only part.
All right, so if it's not just raw size, and it's not just relative size, then what is it? What makes the human cortex so powerful? To find the answer, we have to stop looking at the big picture and zoom way, way in. The secret is in the microarchitecture, the tiny details of how it's all put together.
And right away, we hit something that seems completely backward. The density of neurons in the human cortex is actually lower than in a mouse. You heard that right. We pack our neurons in less tightly. But why? Well, it turns out this isn't a bug, it's a feature. All that extra space between the cells, that's prime real estate for making connections.
And here is the brilliant evolutionary trade-off. By having a lower density, each single neuron has the room to branch out and form an incredible number of synaptic connections. So think about it. We might have fewer neurons crammed into the same tiny space, but each of those neurons is a superconnector. The result? A network that is exponentially more complex and powerful. But it's not just about the wiring. It's also about the types of cells. In primates, about a quarter of our cortical neurons are inhibitory interneurons. In mice, it's closer to 15%. So what does that mean? Think of these GABAergic neurons as a high-performance braking system. They give the brain incredible precision to sculpt and control neural signals. It's not just about hitting the gas. It's about having amazing brakes to allow for subtle, fine-tuned control.
You know, this hunt for what makes our brains unique isn't some new fad. It has been the holy grail of neuroscience for more than 100 years. And this quote from the father of it all, Santiago Ramon y Cajal, just says it perfectly. He was searching for those enigmatic, strictly human neurons, the biological hardware that makes us who we are.
Okay, let's pull all these threads together. What's the final verdict? Well, it's clear there's no single magic bullet for human intelligence. It's not just one thing. It's a winning combination, a perfect storm of several key evolutionary features that all came together.
All right, for your exam, this is your key takeaway slide. Lock this in. The human advantage is a recipe with 4 main ingredients. 1. A big brain with a massive folded cortex. 2. Low neuron density. 3. A super high number of connections, or synapses, per neuron. And 4. A rich diversity of cells, especially those inhibitory interneurons for fine control. It's the synergy of all these things together that creates the magic.
So, I want to leave you with one final thought to chew on. We've seen that the real secret isn't just the number of cells, but the complexity of the connections between them. So what happens next? As our technology gets powerful enough to map the entire human synaptome, every single one of those trillions of connections, what are we going to find? What new frontiers of the mind, of consciousness itself, are we about to discover? That's the exciting future of our field.